And now I'm happy to introduce Dr. Jeroen Dudink from Wilhelmina, um, uh, from the Wilhelmina Kinderzirkenhuis in Utrecht,
Yes.
and or you can also say University Medical Center in Utrecht. Jeroen is a neonatologist and very coming neuroscientist. He's associate professor in neonatal neurology, and he's done a lot of very interesting work on magnetic resonance imaging, and he has also a great interest in the neonatal cerebellum and sleep. And that is the reason why we invited him to come here, and we look very much forward to hearing you.
Thank you so much.
Welcome. Yeah.
Well, thank you so much for your kind introduction, and thanks to the organizing committee for giving me such a warm welcome. I feel very honored being in the presence of so many great speakers. So hope I can do you justice. Well, I'm going to talk about sleep, and I am actually going to repeat some of the things that have been said by the previous speaker. But let me start off with telling you why I got so interested in sleep. And the reason I got so interested was actually because of a baby of 24 weeks that I took care of when I was in Rotterdam about 10 years ago. And this baby was doing quite okay. The only thing we noticed that there was a cerebellar hemorrhage. And the parents told me when the baby was a little bit older, when the child was a little bit older, that they were happy with how it was moving, they were happy with the eyesight and with the hearing, but they weren't so happy about the cognitive functioning. They actually told me behavioral was a quite severe problems in the behavioral region, almost autistic-like. And then I said, well, how can a part of the brain that is so far away from the prefrontal lobe, how can a part of the brain that's really in the back injure or at least damage or alter a part of the brain that you need for communication and concentration and executive functioning? It is really right in the front of your brain. So how is this connection, and what is the missing link? And could maybe altered sleep be the missing link between cerebellar injury and later cognitive problems? And so that was based on a few mice study I read, and so I thought it would be really nice to actually study sleep in infants. And so I began reading about sleep, and well, I guess you all know that we sleep a lot. We sleep about 1/3 of our lives. Well, you should do anyway. Maybe with on calls it's going to be a little bit more different, but you should sleep about 1/3 of your life. And as an adult, you sleep about 8 hours, of which 2 hours of REM sleep. As an infant, as a young child, you sleep almost 16 hours. And if you're a preterm born baby, you sleep even more. And you all know Piaget, who said that play is one of the most important things for a healthy brain in children. But you also know Prechtel, and he said, for babies, it's actually sleep. Sleep is the most important brain activity in babies, and it's the place where development takes place. So we know that we all sleep a lot. We also know it's necessary. And one of the other things that I find really surprising, that sleep is needed for all animals. So tiny animals like fruit flies, which you can see over there, but also the larger animals like elephants or giraffes. And what would be the negative side of sleeping? I mean, what would be, so it's a question for you. So what can't you do when you're sleeping, or why be sleep for the giraffe be not beneficial? Very good. Yeah, so even though there are a lot of threats, and even though you cannot sleep, you cannot procreate, you cannot do any locomotion, well, most animals can't, still we need to sleep, like all animals sleep. And so what do doctors say about sleep? So what do the medical researchers say about sleep? Well, there have been a lot of reports. I think counting up now, you almost reach 20,000 papers on the importance of sleep. And it shows that sleeping has a significant impact on, for instance, your creativity. It makes you happier, it makes you fight off infections, it protects you from dementia and stroke. And maybe the most impressive thing is it makes the size of your testicles a lot bigger. So to conclude, I think that sleep plays a vital role, and else it shouldn't play a vital role, then it's a really huge mistake evolution had made, because all animals sleep, and if we don't sleep, then all organs are affected. Now, before we go on, I think it's always good to give a definition. So how would you, in a few words, define sleep? If sleep, the definition of sleep would count for humans, but also for the fruit fly. So what for a fruit fly or for a giraffe or for a human being would be the definition of sleep? Maybe less responsive, I guess. That's true. So it's a resting state, absolutely. What else? Again? It's a recovery state, yeah, so less responsive recovery. You should wake up, right? Because unlike coma or being dead, I mean, you can actually wake somebody up when they're sleeping, so it should be reversible. And I guess in all animal species, if you have a lack of sleep, you recuperate. Not completely, but you do recuperate. So you actually have an homeostasis mechanism that if you had a very rough night in your hotel last night, usually probably tomorrow, the day after tomorrow, when you're back home, you spend, if you don't set your alarm clock, you spend a few hours longer in bed. And so that's the definition of sleep that we hold true for most animal species. Now, do you need EEG to study sleep? Is it absolutely necessary? For instance, there's a lot of fruit fly research on sleep, and a lot of baby and a lot of fetal. And so do you need to have EEG? Well, basically, you don't even need a cortex to sleep. So no, you don't need EEG per se. It does give you some very interesting insight. But you can use sleep studies, or you can do sleep studies basically based on behavioral research as well. It is very nice that these behaviors have a link with EEG. That's brilliant. But you don't need EEG per se. Okay, so a little bit more to set the background of this talk. You all know that life has a rhythm. You know, because every day the sun comes up, and life, when it started on Earth, it was under the influence of sun. And one of the first people to recognize this, to recognize that all living things have a rhythm, was actually a Frenchman. And it was not a biologist, he was doing physics. And as you probably all know, when you're doing physics and you have to hand in a report, a thesis in this case, you make long hours. And this guy as well, he worked through the night. And what he saw is something very interesting. The leaves on this varan that he had, this special plant, which actually has open and closed leaves during day and night, he saw that they changed. And he said, let's do an experiment. And the next night he actually took it in his cupboard. And he just watched, and he saw that even though there was no sunlight, the leaves opened and closed on a 12-hour rhythm. And he thought, that's interesting, but maybe there is some noise or some... So he even closed all the windows, and he taped all the windows. And in the end, he saw that this leaf, this plant, had a 12-hour on and on rhythm. And he said, so this is interesting. It seems like rhythms are part of life. And we also have a clock in our brain. And in our clock in our brain, it's actually right in the center of our brain. It's here. It's in the nucleus, which means a group of cells. Supra, which means above. Chiasmaticus, it's the part of the brain where the object tracks cross. And so just right above that area, there's a group of cells, and these are very peculiar. And here it's played really fast, but they have a cycle going off and on every day in the same rhythm. You can even take one of these cells and put them on a petri dish, and it will go on and off in the same rhythm. But like a watch of your grandma or grandpa that is not quite working really well, it's not set at the exact same time. And so they wanted to do an experiment around the 1950s. They wanted to do an experiment seeing if this clock has a strict rhythm or not. And you cannot put, like the Frenchman, he could put the plant in a cupboard. But where can you put a human being? Where there's no light, no temperature changes, no sounds of traffic or airplanes, what would you do? The Arctic Circle, but what they did is actually a place, because this was in the US and they couldn't leave the US. Cave. Ground, the cave, very good. So maybe now it would be frowned upon, but this professor took his young student and they went into a cave, like a really deep cave, Kleitman and Richardson, and they said, we're just gonna fall asleep whenever we feel like it. But the other is gonna do the observations. And they saw that the clock, that nucleus suprachiasmaticus, wasn't actually time set every day, it had a runoff. And so there must be something in your brain or something controlling the clock, and actually like your own grandma's or grandpa's clock, changing the settings every day a little bit back. And in our case, it's melatonin. So it's actually the sunlight that has a big influence on there. And how does melatonin does that? How does sunlight do it? Well, we have to go back a really long time ago. So does anybody know when the first bones were discovered, or well, the oldest bones of the Homo sapiens? Any idea how old Homo sapiens are, so us as a species? Who goes for more than 10,000 years? Not according to the Bible, it's 5,000 years, but if you sort of go with carbon dating, it's 300,000 years according to a Nature article. So it's 300,000 years, so that's a long way. But animals that have been around for a lot longer, 250 million years, that's a lot longer than we have been on Earth. They have a special thing, and they call triop. And why would they be called a triops? Any idea? Very good. So there's 3 eyes. Three ops eyes. So it actually has 3 eyes. And the middle one, we have that as well, only in our case, it's sank to the middle of our brains. Now, any idea which gland or which eye is in the middle of our brain? We just got one of them, but it's just not on the top anymore. It's actually sank down in the middle. Pineal gland. Yeah, so we have a pineal gland, and actually these triopses are still around. I saw that they got a flying tiger store next to the hospital, and I think they even might have the triopses there. You just buy the eggs, you just throw some water on there, and they come out and they become about 6 centimeters long, and you could really study how light affects them. But you can't give it to your children. But we have this pineal gland, which is here. So you got your nucleus suprachiasmaticus here, and you got your pineal gland there, and that's the Zeitgeber. That's the one that sort of sets your watch every day. It also means that you can actually give light to your brain and reset your watch in a strange way, and that's lighting. And everybody has a mobile phone, I guess, and everybody has an iPad, and when you watch your mobile phone and watch your iPad, you're telling your brain reset the whole thing. And it's not dark, actually. But that doesn't make you sleep alone. So you got this nucleus that makes you asleep or feel asleep, but there's something more that makes you asleep. And you notice that because if you were on call and if you come home, the light is out. So you shouldn't feel really sleepy because your clock is telling you it's day. But something else actually makes you really tired, makes you want to go to bed. Any idea what the second pressure is on your brain to go to sleep? And it builds up and builds up. And it's all these sort of, you can call them neurotransmitters or leftover materials, and one of them is adenosine. And so adenosine is one of these chemicals that actually builds up if you're awake. And it starts building up the moment you're awake and the moment you're doing mental activity. And then there's one chemical that actually takes the same place on the receptor as adenosine. It throws it away, and you don't feel as sleepy anymore. What chemical is that? Coffee, caffeine. So caffeine actually throws it away, but then again, you have this build up and build up and build up and build up, and then you have to drink more and more coffee to just push it away, and in the end you lose the battle, and then you feel really tired. Okay, how long does caffeine work for? What's the half-life? It depends on the person, very good. But on average, about 6 to 7 hours. So it also means that you have to be really careful drinking if you want to go to bed early. You should stop drinking coffee, at least, let's say, at about 12, 2 o'clock. No coffee anymore. Okay, so a question for you. Who has seen this movie? Very good. You're all very young, so you probably haven't seen it on in the movie theaters, but it was actually, I think it's even on Netflix. And they describe a doctor-patient relationship of a patient that woke up after, I think, 30 years or so. So this patient had a disease. Who knows which disease the patient had? No, not Parkinson. No. It was actually encephalitis lethargica. It's a common description of brain injuries. And what happened was, and it could be that was, by the way, this patient had a type of Parkinson, but not typical type. But in this case, this disease was described in 1930 already. And it said these patients have a lesion, and they have a lesion in which they become very sleepy. And the lesion is actually on the back, so it's more likely to be here. And so from Economo in 1930, and it's a really nice article, by the way, described that you seem to have two centers, a center a little bit here and a center a little bit there, very close to the brainstem. And you have a center that actually promotes wakefulness, and a center that promotes sleepiness. Right? An injury to one of these sides can either cause you to be very sleepy or to be very awake. And now you've got two systems in your brain, and they're also called flip-flops, like a flip-flop switch. You're either asleep or you're awake, and depending on which part is dominating. So you have an arousal system, and you have a sleepiness system. Okay, I leave that to the other speakers, but I just wanted to mention it because repetition is key to understand how sleep works. But let's go back to the roles of sleep. Why do we sleep? And I guess it's very important to realize that depending on the species that we are, you have different roles of systems within your body. And I like to compare sleep with, for instance, respiration. And if you compare respiration in different types of animals, for instance, in lower life forms, which are a lot older than us, let's say in typical worms, then respiration is just a gas exchange. And so it's just, you have your respiratory system just to get oxygen in and CO2 out. But for me, it's different. I'm doing different stuff with my breathing. What am I doing different? Talking, very good. So I'm talking to you using my respiratory system. And if you're working on an EQ, and you also know that a respiration can do something else, or in an ICU, or the acid-base balance, right? So it's also to restore. If I started running now, and I'm not really trained, but I go up and down the stairs a couple of times, and I start like respirating really hardly to compensate for the lactic acidosis buildup, right? So I'm using my respiratory system for different roles. And with sleep, that's the same. And probably we have different roles of sleep than some animals do. So one of these roles is actually related to the following. Who went out last night? I'm not going to tell probably, but yeah, well, a little bit. Okay, and if you went out really early, or if you started running really early, you might have seen these things, right? Sort of, if you go to a city early in the morning, there's a cleanup crew middle of the night cleaning the city. Well, the same basically happens in your brain. However, most of you, I think most of you, have textbooks of biology and medicine that look like this. So there's a lymph system in your whole body except for one part, which is the brain. So how do you clean up stuff if you don't have a lymph system there? And that's where some really nice papers came out, including papers from Nedergaard, showing that during the night, there's actually these pores that open up in the blood-brain barrier, letting in fluid and sort of taking away all the messy stuff. And one of these molecules that has been that is taken away is beta amyloid. And what disease do you associate with the buildup of beta amyloid? Alzheimer's disease. Yeah. So it could be a link there. Okay, so sleep is important for cleaning up, for restoring your body. Sleep is also very important for memory consolidation. And I'd like you to do the following thing, which they did to a group of students. I'd like you to take your hand and number your index finger 1, and then your middle finger 2, and then 3, and then 4. And I'm going to tell you numbers, and you have to, let's say if I say 1, you have to take your index finger to your thumb. And then 2, 3, 4, right? Okay, so 2, 3, 4, 2, 1, 1, 2, 4, 3. Okay, so it starts to become more difficult and difficult. And what they did is they took a group of students. Basically, they said, here's a group of students, and we're going to teach you how to do this really fast at 8 o'clock in the morning, and we're going to retest you 8 o'clock in the evening. And here's a group of students, and we're going to teach you at 8 o'clock in the evening, let you sleep, nice 8-hour sleep, and then retest you at 8 o'clock in the morning. So no sleep, just during the day, and sleep. And it was really surprising that there was a significant difference in how you learn. So this group was actually far better, because they had an 8-hour sleep period in between the learning sessions. So for motor learning, sleep is very important, and it holds true not only for motor learning, but also for a lot of memory tasks. And there is one thing that a lot of students do, especially on a Thursday evening, that completely disrupts this. Drinking. Yeah, and so, I mean, maybe it's not the best advice, but if you would advise somebody that really wants to drink and is allowed to drink at 12 in the afternoon, because then the alcohol is out of your body by the time you go to bed. Okay. And so when does this memory consolidation happen? Well, if you look at a typical EEG of, let's say, an infant that's a little bit older, then you would see these different states of brain activity. One of it is wakefulness. Who thinks, just raise your hands, who thinks that wakefulness is, let's say, this one? Who think this is wakefulness? Okay, then who thinks wakefulness is this one? And this one? Well, some of you didn't raise their hand. Actually, if you look at the typical EEG recording, depends on who you record and where you record, but you got this, I'll show you in the next slide. So, interestingly enough, what a lot of people think, and you're very smart, so you didn't think this. But interestingly enough, what a lot of people think is when you see these really big waves, or really slow waves, that it's a sign of a lot of activity, and it's actually not. It's actually, this is a non-REM stage sleep 4, where you see the slow waves coming up, and this is actually really linked as a sort of retransfer of your brain. So getting the information for your hippocampal area to your cortex. It's almost like that's the time where you process the information and it's sent away. So I think REM sleep, you could compare it with the LinkedIn of your brain. You're just linking and you're making connections and you're being creative. And non-REM stage 4, you could compare it with retransfer. It's stored. So if you take out non-REM 4 and you teach students stuff, definitely not going to remember it for a long time. And what Philips did and other peoples did, and it's based on a few papers that came out recently, is they made machines now. They started making machines actually trying to hack sleep. So what they did is when there's a slow wave, just give a tiny electric shock or a tiny sound impulse to make the waves bigger. And that's interesting because if you get older, if you get over 40, if you get over 50, and especially sort of over 60, over 70, then your slow waves decrease. So, and your memory capability decreases as well. But if you enhance the slow waves, you can actually upgrade your memory by about 30%. And so Philips thought that it was a really good idea to make these machines. And they're out for like 2 months now. But you can buy, you can put on your head, and they give a small stimulus at the time of a rising curve of the slow wave, and thereby you can hack sleep. There's also, so that's the second role of sleep. It's memory consolidation. It's cleaning up the system, and it's memory consolidation. It's also a really good way of getting rid of all the weak connections, because probably most of what I'm telling you, you will forget, and that's okay. But so it's also good, but you heard it anyway. So it has to be out of your system, because tomorrow somebody else will say something way more interesting to you that you want to remember. But you have to eliminate everything, or hopefully not everything, but a lot of what I said to you. So eliminating weak connections is also a part, and this is studied extensively in fruit flies. Now, how would you study sleep in a fruit fly? So how can you do this? Well, one of the things is you can put them on a sort of treadmill, because you can put them on a tiny ball with some air underneath, so it's really easy for them to walk around in every direction they would like. And so you can have movement, and they take tiny shakes of their body, and you can record these tiny shakes, but they shake less at the moment they're in sleep, and they actually do have quiet sleep and active sleep and wakefulness periods. If you keep them awake with noise and with light, then they sleep longer afterwards. So fruit fly is really nice. And if you keep them awake by being really mean and you keep them awake, so they're really sleep deprived, you see there's less elimination of the connectivity. And so these experiments, well, they might give an indication that sleep is also important to get rid of the excess stuff. Then there's some people that saying, well, sleep is very good for restoration. Sleep is very good to get everything that has been depleted, like the battery that's been depleted in your single neuron, on single neuron level. It's good to sleep. And imagine that we would stay here for the next couple of days in this room. Small toilet here, but you're not allowed to leave. I'm going to give you some food, but you're not allowed to leave here. It's a lot more easier if I would say, okay, guys, now it's time to go to bed, than if I would say, sleep whenever you like, right? Because if we all go to bed, then there's no chatter, there's no noise, and so sort of having these slow waves gives a lot more rest, some researchers think, than having activity going on all the time. So it's really nice paper in Nature, Reviews Neuroscience. So we talked about restoring function of sleep. Now another role that has been overlooked by many doctors, and that is that sleep actually is very good for your immune system. And again, they did this really incredible experiment where they say, we're just going to make a few groups. And last time I gave you no sleep, so I will give you a lot of sleep. But they said, here's a group of people, you're going to get 5 hours of sleep, and then we're going to wake you up. And you can have 6 hours, 7 hours, and you can sleep like how long you want. And then the next morning, they injected a little bit of rhinovirus, which sounds really bad, but just a cold, in everybody's nose. And they looked, who's going to actually get the symptoms of the cold? And these people were fine, like you were okay. You had 8 hours of sleep, but about half, almost half of the people in this group got the symptoms of the cold. And so sleep is actually so important for your immune system. And that's all fun and games when it's all about a simple virus. But there have been many studies showing that your natural killer cells actually reduce their activity by about half if you sleep 5 hours or 6 hours. And what do natural killer cells do? They're the ones responsible for finding tumor cells. They're the ones responsible for finding that odd cell out. They're your James Bond cells. So it actually protects you from the risk of having horrible diseases. Okay, but all this just to show you the different roles of sleep, and there's also another role, and we've been getting some information about that by the last speaker, sleep and brain development. And I think one of the articles that I can really recommend is an article that started off a lot of important work. And it's an article by Rolf Wark et al., showing that it cannot be coincidence that the older you get, the less REM sleep you have, the less sleep you have. So there must be a role between sleep and brain development. And it actually made a good case for this. And so it actually sort of set off a lot of studies that were already ongoing about the importance of sleep, but sleep is not there for nothing, right? So sleep is important for brain development, and this was, I think, one of the articles, at least pushing everybody in the right direction. Okay, so how do you study sleep in infants? We talked about EEG, and we said, well, it might not be extremely necessary, but it could be very interesting. But if you show an EEG to a specialist, they're completely fine. But if you show it to most people who are into adult electrophysiology, they probably say this baby is brain dead, or at least has some very strange way of showing cortical activity. And so it is a challenge to study EEG in preterm infants. And instead of having this clear 4-stage non-REM and REM stage, and then a normal brain activity, you might see something like this. So it's sort of a burst of activity and then nothing for a while. And so that's why it is a speciality on its own, and you need people to actually interpret it, experts to interpret it. And then if you get more into it, you see that when the brain organizes itself, you actually see that stuff happening week by week. And you see, for instance, that the periods of bursts of a lot of activity, the periods in between shorten. You can see different things when babies are sleeping and when they're not sleeping, and you can use EEG, I think, as a good marker for brain development. So if you have a weekly EEG and you would see the sleep stages and you see how it's organized, it can actually tell you, and it's been shown in a lot of articles, it can give you a good prediction of long-term outcome. Just because you need a healthy brain to produce healthy sleep. And also you need healthy sleep to produce a healthy brain. Another thing that has been really nicely shown, and there's a lot of articles about this, especially from Samsa van Altalo, showing how if you reset your filtering settings, you see another phenomenon which is really specific for these babies, and it is a pattern which is called spontaneous activity transients. And there are multi-band events, and again, you see that progressing during the different weeks of gestation. And it's also really nicely related to the formation of connections between the thalamus and your cortex. So again, it's a really nice mirror of what's going on in the brain itself. And then using this stuff and knowing a lot about EEG, you can actually see how sleep and wake cycles change within the maturing brain. But what if you don't have EEG, for instance, in a fetus in the abdomen? When the fetus is still within mom's womb, you have to use other methods. For instance, you can do an ultrasound and look at heart rate, eye movements, body movement. Again, if a preterm if a baby is born preterm, you can also use the heart rate, the breathing rate, the muscle tone, all these behavioral aspects of the baby that can give you a clue on whether the baby is sleeping or not. And so one of the things, if you study this, that you see that, I'm going to call it REM sleep, but it's actually active sleep. So active sleep is far more common in babies than quiet sleep. In adults. And so it's almost like they're playing their own Mozart. It's almost like they're generating their own endogenous activity to organize their cortex. And so that's very important, I guess. So when these things happen, that's also the period where the brain plasticity is the most, when the most new synapses are being formed. And also, so I think, yeah, I can really concur with the previous speaker that it's vital for brain development. Now we wanted to study this because we wanted to study, really wanted to study how the cerebellum affects sleep. And so I said, let's go on the internet, let's find the sleep score and just use that, and then we're there. And then I found that there's not one. There's actually many. And these are common ones, but we counted up to, I think, 20 or 30 sleep scores that are around. And so that's really difficult. Which one do you pick? Describing wakefulness, you got like different people describing or using the wakeful state. Some say state 1A, 2B, one. So that and the same for transitional stages and sleep stages. So what we did is we took we just tried everything basically. We said we want a system that can discern active sleep, quiet sleep, intermediate sleep, and wakefulness. And we weren't really interested at that time at the painful procedures or whatever. We were just sort of, I was actually really interested in quiet sleep, active sleep. And so what we did is we just build up, based on an article of 2060 of Wert et al., we just build up our, how do you say that, a stepwise approach. So instead of just saying like these are the features that you see in active sleep, we said, okay, first thing you do is you walk to the cot. Second thing you do is you note this down. Third thing you do is you look at the eyes. Are they open, are they closed? So we do the stepwise approach, and we did it over and over again until we had a system that we could train somebody new within a few hours based on a few movies, and they would score very highly so that there would be a very good inter-rater agreement. And it took us like a year or I think a year and a half to do this. And so every time somebody new comes in, we just grab them and we say, please come here, please follow this and score. And see if you score the same thing as us, because there are behavioral experts, and they're far better than we could ever be. But I wanted the system that's written down on a card that everybody can use, that the behavioral experts agree on, but also that can be learned very easily. And so we're in the process now. At the moment there are 3 students who've just been trained, and I asked them, what is your latest inter-observer agreement? And they said, well, we're still very good at active sleep and quiet sleep, and you can see it's called a coherence kappa between observer 1 and 2, 1 and 3. So they're okay with active sleep, they're okay with quiet sleep. It becomes difficult when you record intermediate sleep, for instance. And so, and based on their results, they do it again and they test it again, and sort of it's an up and down, and we're almost getting there. And so basically what they do is they just sit there, they record everything, they just sit there and they look at the babies, and they notice, for instance, that this baby is a very quiet sleeper. Oh, sorry, this baby is a very quiet sleeper. And here you see some tiny movements, and then every minute they record. The last minute was this. The minute afterwards was this. And then they sit there with the 3 of them, and then they compare. What did they score? And so in the meantime, while they're doing all the hard work, it's my job to sort of think about other stuff. And one of the things I noticed that it would be so nice if the baby wasn't hooked up until any wiring. So it would be very nice if we have a system that can actually monitor the baby's sleep through the incubator without wiring. That would be perfect, right? Because then you have no things attached. And so I came across this company who made Bluetooth-like sensors. And you all have Bluetooth on your phone, I guess, right? And so Bluetooth sends out a tiny signal that is very harmless, trust me. But Bluetooth sends out a tiny signal, and you can actually send that out in pulses. And the nice thing about Bluetooth is, if I would hide here, I could still read your Bluetooth, because it just goes through everything. But then again, it would bump until this and come back. And so if I would aim my sensor at you, then it would bump until your chest, it would go through your chair, but also bump back. And I could see your heart rate, and I could see your breathing rate from it. So basically, if I would be really mean, and I would want to know if you're paying attention or you're paying attention, you can just aim my device, which is about this big, at you, and see your heart rate, and see your breathing rate, and see if you're paying attention or not. But what happens then is you can actually, what we did is we took that tiny chip, it's called the see-through. We took the see-through chip and we put it next to the beds of babies that got polysomnographies, or well, children that got polysomnographies. And we said, let's make this the gold standard, and let's see how our device is doing compared to a polysomnography, let's say, breathing pattern. And we saw that it was quite accurate. And if you do that, then you need to do something else. And here is where it always becomes very difficult for me. Because then you have a signal, but then you have to decide, how does the computer learn if it's sleeping or wakefulness, or how do you do that? Which machine learning tools do you use? And well, I'm not an expert here, but that's why working together with technical universities is so nice. Because they said, well, let's go for the support vector machine learning. And I believe them, because you could have showed me anything. But if you do support vector machine learning, and basically to explain this is that you have two states, or you have two things, let's say a dog and a cat, and you want Google to select all the dogs and select all the cats. But some dogs look a little bit like cats, Chihuahuas, and some cats look a little bit like dogs. You know, you got these big cats, and so it's really difficult to place the line where what is dog, what is cat. And basically, support vector machine learning is that you have two sleep states. Some are very clearly different, some are a little bit, and so it's sort of, it founds the boundary between these two states. And if you start doing that, you actually come across rather a good, just based on this Bluetooth device alone, you actually come across quite okay accuracy of sleep states. And this is in older children, but we're now trying to do this in younger children as well. So ideally, you would have a system built within your incubator, of near your incubator, and this really nice image I took from the paper of Huerta et al., showing you in which state the baby is, but also showing you if there's anything elective you would like to do. It could tell you, please wait 5 minutes, because then actually the chances of getting out of your active sleep are very high, and then it's actually in a wakeful state, we predict this, and then it's okay to do. And so that's what we're aiming for, and that's what our research is based on at the moment. But I wanted to talk about the cerebellum. And so let's go, for the last few minutes, go back to the cerebellum. I haven't found the answer yet, but what I did notice when I started reading on cerebellar injury and outcome, it is actually quite convincing literature that cerebellar injury early in life is actually a major predictor for having cognitive problems or behavior problems. And this paper came out in 2014 showing that what are your chances of having autistic traits? Well, the highest chance of having being autistiform is having an autistic identical twin. The second highest, and it's way there, is have cerebellar injury at birth. Far more likely than having a mother or a father with autistic tendencies. And so that is really, it gave me a lot of insights that, okay, so cerebellar injury leads to problems in cognition and attention. And we did the systematic review last year, it was published in Pediatrics, showing that cognitive problems, well, mostly cognitive problems, if you see the cerebellum in the middle, so the middle hemorrhages that actually are either bilateral or in the middle of the cerebellum, they actually are most likely to cause these problems. And who knows a lot about the cerebellum? Probably a few of you. So if you look at the cerebellum is connected with almost all brain parts, right? So you have your brain, and then you have, of course, output of your brain. A copy is of your output is sent to the cerebellum. Whatever you're doing is going back, and it's like there's this coach telling you, you did it all wrong. Or you're actually quite good. It's fine, you did what you're supposed to do. So your cerebellum, I like to see it as a sort of a teacher of your brain. And that's for motor, but actually cerebellum is a lot more than motor functioning, because if you have an injured cerebellum, you also see, well, the amount of papers out now is astonishing, but you see these problems in all these different areas. And why is the middle so important? Well, I guess that's because if you look at the cerebellum, you have, like, how many brain cells do you have? Maybe if you went drinking a lot, it's a little bit less, but let's say you got about 90 billion, so 89 billion brain cells on average as an adult, right? And where are they housed? Well, 2/3 of them. So most of them are in your cerebellum. So your cerebellum is actually filled with a lot of neurons. And all right, they're tiny and they're mostly like the granule cells, but there are a lot of granule cells. They're not always active, but there are a lot of them. And they project 100 and 1,000 to 1 to these Purkinje cells. And so there's quite a convergence of all these granule cells, like almost all cells in your brain, so that they convert to this Purkinje cell. And then for every 40 Purkinje cell, they go to one output cell, one deep nucleus cell, which is in the middle. And so it's not surprising that you have an injury here, you're okay. I mean, you can spare a few. But if you have an injury in the middle, where your output cells are, where your deep nuclei are, then you're in trouble. And that's exactly what happened in these children that have a midline problem. Okay, so did anybody study sleep in the cerebellum? Yes, they did, and it was quite some time ago as well. So in 1974, they did studies in which they took needles and pinned them in the cerebellum. And you won't see that now, but who knows this technique of radiography? Does anybody know? Why can I see? It's an X-ray, and I can see the ventricles very clearly. And if I take an X-ray of your brain, I don't see any ventricle. So how can? Yeah, it was actually air that they injected in the liquor. So they inject a lot of air into your brain, and then you can really see and that's how they did it, right? 1974. That's the technique. Now we have MRI scans, but the first MRI scan on a human was in 1973, and that was just the first one. So, I mean, we have improved. But it allowed them to visualize where they thought the cerebellum would be, and they would stick needles in. And then you would actually see that if you stick needles in some parts of the deep nuclei, you could actually see these signs. And it's not EEG, it's actually the EEG of the cerebellum, so cerebellum graph. But you actually see signs of sleep and wakefulness within the cerebellum. And I mean, basically, I would have liked to tell you more, but we're doing research now with Kanto, who is from the same university that produced the heart rate variability measurements devices. So the Free University of Amsterdam, and working together with her now to set up a mice model to see how injury to the cerebellum leads to differences in sleep, which can lead to differences in the cortex. So to summarize, I think sleep is very important for the brain development. And what needs to be done is actually go back to behavioral observational studies. So you see a lot of papers came out in the 1970s, 1980s, and then it stopped. But we need to go back. We need to do a lot of observations, because linking observations is the only way, linking them with modern techniques, is to get sleep quantified. And also we need neuroscience experiments to unravel the complex relationship between sleep and brain development. I hope I didn't bore you too much. I hope you're not too tired. So, but I would like to thank you for your attention, and I would like to thank the research team for supporting me in this.